I'd like to introduce you to Lecture 14, which is Chapter 11 in your book on nerve cell function. And this is going to be a very um, difficult concept to grasp, but if we just look at some basic principles of how nerve cells work, we should be able to get through this. So when we look at neuron function, what is a neuron, or what we sometimes call a nerve cell? Because we have to be careful, because we're going to see that there are, two major types of nerve cell. So neurons are what we call irritable or excitable cells, and it just means that they carry what's called an electric current in them, and pretty much think of them like a battery that when you connect it to something produces an energy flow, and many things can cause these cells to become what we call excitable. So a nerve cell is doing its job when it releases this electrical energy or releases its potential in what we'll call an action potential. Typically, nerve cells are initiated by a stimulus of some type, and this produces again what we call an action potential or a new impulse. This stimulus is usually due to a chemical signal called a neurotransmitter. We will see, but other things like a direct hit or varying concentrations of salts can cause an active potential. We're going to see that each nerve cell has its own nerve impulse. That means a nerve cell does not have a graded response. That means if you feel a little pain or a lot of pain, it's not due to one nerve cell doing that, but rather nerve cells that send various signals to the brain of either being a lot or a little, and sometimes it has to do with the amount of nerve cells that are stimulated. Again, we're going to look at this idea called potential difference because nerve cells again work by voltage, and we're going to look at the concept of current, which basically in nerve cells is a flow of electrical charge, or what we call charged particles or ions. Now, the whole secret of a nerve cell is its membrane, which is sometimes referred to as the neurolemma. And there's various debates about what is actually the neurolemma of the cell. Okay, so we're going to see that the cell itself is going to, that membrane is going to separate an electric charge difference between the outside of the cell, or that means the fluids around the cell, and the cytoplasm inside the cell. And because of this difference, we're going to see in the concentration of ions inside and outside the cell and electric charge, something called leakage has occurred. That means leakage just means that naturally charge and ions are going to travel back and forth across the cell through these special channels. And the cell is going to correct this to what we call gated channels that are going to allow the cell to compensate and keep the cells in what's called a resting potential. So don't worry so much about memorizing these types of gates, but just know, and channels, just know that they're there to carry out a job, that these are membrane proteins that are letting the cell produce an electrical charge and kind of store an electrical charge. So now just let's briefly look at these channels. And again, these are just membrane proteins that act like barriers and gates that selectively allow either something like sodium to go into the cell or potassium out. And sometimes these channels open to allow a little leakiness. That means exchange. So sodium can rush in, potassium can rush out, depending on the concentration gradient. And then we also have what's called these voltage gated channels. And these are going to be the important ones in looking at how nerve and actually muscle work. Okay, so what happens with nerves is when we get electrical stimulus, I mean something that causes electrical stimulus in the cell, that causes these channels to allow or block sodium. So when an electrical charge goes through a nerve cell, sodium is to be allowed to go through these channels. I'm going to see that there's other ones that allow potassium to go out. So there's two types of channels, what we call these little kind of leaky ones, which under certain conditions allow sodium and potassium to go back and forth to kind of keep the cell somewhat chemically stable, because or I should say in a certain degree of instability that's very important for this cell to be like a charged battery. And then these are going to control actually the electrical flow that allows a neuron, a nerve cell, to communicate to other neurons or to muscle or to various glands or organs. Now, when we look, now think of this as being a nerve cell, because we're going to, and there's your nerve cell. So we're looking at this thing called an axon on the nerve cell. Okay, so the inside of the nerve cell is a cytoplasm, And this is just showing now that it's going to be a negative charge of about what's called minus 70 millivolts. That's a very small charge. That's 70 thousandths of a volt. Okay. The outside is going to be what we call relatively positively charged. So when we look at what's called a resting nerve, think of it as a charged battery. Then when you connect that battery to something, an electric charge comes out. So this battery right now is resting. That means its normal charge it has a negative component on the inside, a positive on the outside, just like you see on batteries. And what we're going to do is kind of allow a connection to produce electricity. That means we can allow positive charges to flow into the cell that produces electricity and negative ones to flow out. And that's going to produce an electric current that we're going to see affects that minus 70 charge. So don't worry about what the minus 70 means. It just, that minus 70 though is going to be an important number a little later, because for a cell to actually do its job, it has to be pushed out of what's called that minus 70 resting range to a certain minimum level before that cell actually carries a current. Very much like hooking up a battery to something. You can hook a battery up to a certain circuit that allows it to do its job or not, depending on what you're operating on that circuit. So you're going to hear this term resting potential. And resting potential just has to do with what we call the difference between the electrical charge and the ions, sodium potassium ratios, levels in this intracellular fluid. It just means inside the cell. They abbreviate ICF. And we're going to see that the intercellular fluid is going to have low levels of sodium. Don't worry about the chloride. Lower levels of sodium on the inside of the cell than the outside. You're going to see it has higher levels of potassium on the inside than the outside. Okay, and like explained right here. Now, this resting potential, the inside the cell is going to have what we call a relative negative interior. And again, this is going to have to do with the relative ratios of sodium on the outside, potassium on the inside. So we're going to have almost 6 times as much sodium ions on the outside of the cell, which are positively charged. Look at that. And a smaller amount of potassium on the inside, even though there's a lot more potassium on the inside than the outside, but there's to be a smaller amount, which makes, so when you have more positive on the outside, less positive on the inside, that gives the inside what we call a net or a relative negative charge. Again, don't worry about this. Mostly know what is in a large amount on the outside of the cell, a large amount on the inside, and that the cell in resting condition is negative charge on the inside compared to the positive outside charge. So again, when we start looking at the resting cell, a resting cell is not really at rest. What it is doing is taking into account what we call leakage. That means because you have this cell, which has a large amount of sodium on the outside, a small amount on the inside, the principles of diffusion are going to try to force that sodium to go across the membrane into the cell. And that happens through what we call these leakage channels. It prevents disruption of the cell. So it allows a little while still maintaining somewhat of a positive outside, negative inside. But you still have to remove that excess sodium, and that is done by what we call sodium potassium pumps. This is an active transport method that pumps 3 sodiums out for every 2 potassiums that will come in. Because what's going to happen here too is you can see that there's going to be a lot more potassium on the inside than on the outside, and potassium is going to want to try to leak out. But we could allow a little to leak out, but that's corrected. So just appreciate the complexity of this. And appreciate, guys, that by what you do to your diet can force more sodium into the cell than wants to be there. For example, eating a high sodium diet or taking high potassium can actually prevent potassium from coming out, and it can actually make it harder for a nerve cell to do its job. And we're going to see that a little later. So this membrane potential can be broken in 2 ways. That means we can produce an electric current in 2 ways that affects that cell. One could be in what's called a graded way. That means the cell can produce electricity over a short distance in itself. And this occurs in muscle tissue, we're going to see, but also in nerves. In muscle tissue, it could actually cause a minor contraction. In nerve tissue, it might not cause the nerve to do its job, but if you have enough of these coming at each other, it could actually cause a larger transport of this electrical charge. And then there's an action potential, and that means this can be a signal going to a cell that causes an electrical current to travel the whole length of the cell and have the cell do its job. But sometimes with graded potentials, we can get a bunch of little graded potentials that can cause an actual action potential. But graded potentials have a purpose. They're sometimes needed to just maintain the integrity of the nerve cell, and sometimes what they're used for in the body is to send a signal to a nerve cell, but not have that nerve cell do anything until a lot of those signals come and repeat and repeat and repeat. And we'll see that there's a purpose to this eventually. So when we look at what's called a graded potential, sometimes an electrical signal basically travels a short distance from here to the dendrites, sometimes just to the nerve cell body, and it does nothing. It does nothing. So that means a signal stimulates the cell to produce an action potential, and it doesn't really go anywhere. Now, why would you want this? Again, we're going to see that this only works in certain situations where we have other nerve cells communicating with this. And if they each give a greater potential, that can do enough push to cause what's called an action potential, which you're going to see below. So a lot of this is based on what we call distance. Now, an action potential is when a strong enough signal coming in then causes this cell to produce an electrical current that travels down the whole distance of the cell, and you need that long distance to have that cell communicate to other cells or other structures that receive that signal. So don't worry so much about this. But sometimes what happens with your body is sometimes cells accidentally communicate with each other or send signals that shouldn't be, and if those are graded potentials, it doesn't cause a response, and it sometimes protects you from things, some inappropriate movements. But again, let's not worry too much about this. So, what does a resting potential look like? And how does this get to the point of causing what we call an action potential? So a resting potential means is that we have this dynamic cell. That means it's always correcting itself, trying to keep a cell so that there's a lot of sodium or natrium on the outside, positively charged. Very little on the inside. Now know that some is leaking in, some sodium's leaking in, okay? And some is also leaking out in a way too. It just depends. But what we try to do is keep this balance to the point where we're pumping out sodium, okay, at a greater rate than it's leaking in. Now look at all the potassium in here, in the cell. This potassium wants to try to get out, and sometimes it does. So what we're hoping to do is to keep pumping potassium in to keep that level high. So, but this is not showing what's truly there. The concentration of ions on the outside is greater than on the inside. So what this does is it makes this a very big positive charge, this a very little positive charge, which I hate to say this really then produces what we call a net negative. So don't try to understand this as much as we have this negative charge and here's your minus 70 millivolts right there. So we're going to see that this is stable for many cells. Some cells have what's called a minus 80, minus 90 potential. Some cells are a minus 65. It all depends on their sensitivity. So in one nerve, you could have axons, nerve cells that have different variations of a resting potential. Because we're going to see that that minus number is going to be like what we call a threshold. That means we can push that number to be a little less negative, a little more negative to affect how well that cell works. And you'll see that in a little while. So the resting potential, just remember, positive outside relative to the inside. So once we get to the resting potential, what happens if something disrupts that resting potential? That means we're going to see that something happens where that cell membrane is going to be disturbed. And there's various ways you can disturb that resting potential. That means you could force those ions to start exchanging and forcing their way into the cell or forcing their way out. And that produces an electrical current. And what that's going to do to the cell is do what's called a depolarizing. It means it's going to take that positive outside, that negative inside, and change it to the point where the charge is going to reach a point at one time where it's zero. That means it's the same charge inside and out. And we're going to call that depolarization. So depolarization is the beginning of a graded or an action potential. And it's going to be started or defined by all of a sudden, sodium just rushes into the cell. And what the sodium is going to do is try to make the amount of sodium equal on the inside and the outside of that cell, which eventually changes the nature of that cell. It's going to change that ratio of positive to negative charge. Now, what does the depolarizing phase look like? It means if a signal somehow comes to the cell, it's going to affect how these proteins work. So here's these channels. We have what's called a sodium channel and a potassium channel. Now, the sodium channel is going to be very important because they are normally going to be closed. They're normally closed. Now, sometimes leakage occurs, we're going to see in other channels, but they're normally closed. They're maintaining a large amount of sodium on the outside. We're going to see that some type of signal, it means either a neurotransmitter from another cell, some type of chemical, that could be what we call a neurotransmitter mimic. For example, nicotine in cigarettes mimics certain types of receptors that can actually cause a neural stimulation, particularly a graded response in many cases, that just kind of tweaks that neuron. Okay, it could be an electrical shock, like if you stick your finger in an outlet. It could also be a physical thump. That means I actually hit a nerve with a hammer or a metal probe. Or I could even pour extra sodium on a neuron. And that will sometimes put so much sodium out here, they just literally force their way through that closed gate and force it open. So the depolarization, you're going to see the first stage is these doors opening. And that sodium is now going to rush in. That's the first stage of depolarization. And we're now on the way to getting that cell out of that resting potential. And that sodium is going to produce an electric current. So now we're seeing the second part of the depolarization stage. Sodium is rushed in. Look at that now. Lots of sodium on the inside. And what does that potassium do? Because there's so much potassium in here, they tend to rush out. And now the potassium's higher here. And now look at your electric charge. It actually turns positive for a while because what's happening is you've reversed the charge. Okay, and don't worry about this, but this now becomes positive, which means that this cell is non-functioning because it only works in the reverse. So don't worry about what causes this positive, but just know now there's lots of potassium out here, lots of sodium in here because there's so much so much sodium here, so little here, it reverses the charge. So this great amount of sodium is on the inside, so this tends to be more positive than that, making this negative. And again, don't lose sleep over it. We're measuring the inside of the cell. That's what that little probe's supposed to mean. So this is the depolarizing phase. That means the cell is unstable, but it produces an electric current that we're going to see destabilizes the rest of that cell. Because when this occurs, guys, all the rest of that membrane going down off the slide or even in this direction, that causes a mad dash. So think about a riot started here with sodium rushing in, potassium rushing out, and that causes the same thing to occur next door in the next city. And it keeps going down and down, and we call that an impulse. So this is a beautiful thing. But once depolarization occurs, that cell is totally useless. And sometimes what happens when your nerve cells die, they tend to depolarize. And that means they can't be fired. Many nerve cells do that. Now we got to get things back to normal. And this is going to take a period of time. Sometimes we call refractory period. That means, man, we got to come to a rest. Muscle does this, nerve does this. It has to get back to normal. So now we do what's called repolarization. That means the cell now, after depolarization, positive on the inside, negative on the outside, what we want to do is swap that around. And that means what we now have to do is pump all the sodium out, the potassium back in, and get that resting potential. And don't worry about the gates that do this so much, but do know that these gates open and close to do this based on, again, relative charge based on the conditions of the cell. So with repolarization, so now we get these little pumps going on and these little channels basically correcting the problem. So these are now shut. They're not going to allow any leakage, but we're going to pump sodium out, allow potassium to go in, and get this equilibrium back to normal. So this is now almost at the resting, actually it is at the resting potential at -70. So to understand though an action potential, we're going to have to kind of look at a diagram now of how much stimulus does it really take to cause a graded or action potential. Let's just review quickly what this idea of propagation looks like. So here's a resting cell, okay, positive on the outside, negative on the inside, a lot of sodium here, very little potassium, a lot of potassium here. They're only focusing on the potassium and sodium right now. Okay, so what happens, you get an action potential. That means action potential means sodium at first rushes in. That's part one of the action potential. Part two of the action potential is potassium rushes out because it's kind of trying to dilute, dilute itself. Okay, and then what happens is now you're depolarized, and now here we're starting to repolarize by this thing called a sodium potassium pump. Is pumping potassium back in to make a lot of potassium on the inside, and we're pumping sodium out to correct this. So this is called a sodium potassium pump, and this requires a hell of a lot of ATP. A lot of ATP, about 70% of your body's calories, is maintaining basically resting potential in muscle and nerve, and also coordinating an action potential or muscle contraction. 70% of your calories at a person at rest. I think that's kind of neat. So let's look at this from a graph. This graph is called membrane potential millivolts. That means we're measuring the relative charge of the inside of the cell compared to the outside. So a resting cell is -70 millivolts. Now, what that means is that it's stable at this point, but it's going to take a certain amount of stimulus to make it to the point where all these channels and pumps start doing their job. And that's where this number comes in. For most nerve cells, we have what's called threshold. That means a cell, when it's at a charge difference of -55, that means a certain amount of potassium on the inside, a certain amount of sodium on the outside. This charge is all determined by sodium-potassium ratios on the inside and out. When a cell gets -55, that's enough of a stimulus on that cell to make a graded or action potential. So that means you have to add enough energy to get a certain amount of sodiums in and out that go from -70 to 55. Okay, that's all you need to do. So what does that mean? You have to have like a -15 millivolt difference. Now, some cells are not at -70. Okay, they could be at -90, for example. That's their resting potential. Now, why would you want a neuron like this? Because they are harder to get an action potential. That means you have to have a lot more sodium and potassium moved to get to that -55 level. So you have to do a lot more shuffling around to get this nerve to become more unstable so it produces electricity. You can have some that are at -65. Oops. -65 resting, and that means it only takes 10 millivolts difference. So that cell is what we call easily excitable. So in your body, you have cells that are easily excitable, very excitable. And this is called a graded response. That means that means the brain, if it wants you to feel a different type of stimulus, that means either a little or a lot, sometimes what it can do is it keeps track of these nerves at a minus 90, minus 70, minus 65, and it says, oh my gosh, if a minus 90 signal is coming in, that means there's a lot of stimulus coming in, and that's how your body perceives it. So that means a stimulus that could activate a minus 70 won't activate a minus 90. Don't get too confused by that, but this is be very important in A&P2 when we try to understand how organ systems work, and particularly the collaboration between the respiratory system and circulatory system and your heart in general. So what does this look like on a graph? You have a resting potential. Something causes the membrane to become disrupted. That means a certain amount of sodium and potassium are exchanging, and if it exchanges past a certain point where the inside of the cell is now minus 55, all hell breaks loose. And now the gates open, sodium rushes out, potassium rushes in, you get your action potential, and then what happens to that charge? That charge is very positive. Now what's happening is we're trying to correct it to get it back to normal, and we try to get it back to normal, and now we reach, that's repolarization, and now sometimes we overshoot. This is called hyperpolarization. Don't worry about that. It's just a correction factor, and now we try to get the cell back to the minus 70. So during a period of time, that means milliseconds, that means tenths, look at this, thousandths of a second. It takes 2,000ths of a second for an action potential. And this is important to pay attention to because there's a procedure called where we can look at the speed that nerve fibers work. We can actually look at the electrical conductance of nerve fibers and see how long it takes a nerve, and in research, an individual neuron to go through this period. And this tells you the relative health of a cell. Now, guys, what happens, and this is very important, if you take in too much sodium in your diet, it can take the minus 70 threshold and put it about here. And that means your cells are now more sensitive to an action potential. And you can have inappropriate action potentials, particularly when you look at heart and smooth muscle. It can cause them contract when you don't want them to. And this could also cause nerve firing where you don't want it to and produce twitches and other weird body movements. If you have too much potassium in your diet or too low sodium, it could put that cell at a minus 80. And that means now the body is communicating to that cell and thinking, why the hell isn't it firing? I'm getting the right amount of sodium pumped in and the right amount of potassium out. Or really, it's mostly the sodium going in at the first point. And this makes that nerve hard to fire, and you can get an insufficient response. And this could actually raise or lower blood pressure. It could increase or decrease cardiac rate, or it could affect the strength of a muscle or the amount of muscle fibers that are being excited during a movement response. So just be aware of what's going on here and understand the concept of this. So here's a more detailed view of what we call the action potential chart. So there's your minus 70. Something caused that minus 70 to be pushed up to about minus 55. And that means somehow sodium was rushing in. And now it breaks the point where sodium just gets unreleased. And so much sodium goes into the cell and makes the cell positively charged on the inside. That's your depolarization, and that now encourages the potassium to be shooting out because those potassium gates open up, and that then kind of brings the charge back to a minus 70, but it's reversed. That means there's sodium on the outside, potassium on the inside. Sodium on the inside, potassium on the outside. Sorry about that, and you want to reverse that. So that now gets reversed through these sodium-potassium pumps, and the charge kind of sweeps down a little and then goes back to normal. So again, this is this period of healing, that means getting the neuron to get back to the normal where it can get another stimulus, is called refractory period. And sometimes, guys, during this stage, you can get another signal to pass through. If a graded signal, if there's enough of these signals, it could actually produce another action potential before the cell totally goes to rest, and you can get another blip like this, and then another partial blip like this, before it completely gets to resting potential. And don't worry so much about this. This is more important during very intense activities, during very rapid activities. So the propagation of the action potential. It means when an action potential occurs in a tiny piece of cell, it will cause everything next to it to go crazy. So all you need is one stimulus in certain cases, or a certain amount of stimuli, to get that cell to that minus 55 millivolts point. And then once it gets past that point, that means it's now the cell is more positive on the outside, negative on the inside. It disrupts the rest of the cell, and the whole cell acts as if another stimulus is coming in, and it basically stimulates itself. So again, what does this look like? As you get a local action potential, so notice now the outside is negative, inside is positive. That causes these now, the sodiums here, to rush in, potassium eventually to rush out, and that whole instability, that means that electric charge, because now you're producing electric charge here, that electricity literally travels down the cell. And notice that it causes what we call propagation. And that propagation is about a 2 millisecond event for that to occur. So there we go. We got the action potential, it's moving down, the cell has now recovered at that point, and a new propagation begins. That means this is going to move here and here and here eventually in a graph that looks like this. So the electrical circuit is traveling down. Notice that that little unit is going to cause that to occur, and you have a propagation. So how is propagation affected? The speed at which a nerve cell communicates is based on various things. The two major things are the diameter, that means the thickness of that neuron, and also the effects of what's called myelination, that means glial cells that are wrapped around and insulating that neuron. So again, diameter and myelination. And this is why some neurons in your body travel information faster than others. And this is very important. We particularly talk about nerves like, for example, your lumbar vertebrae that travel all the way down to your toe. We have neurons that do that. I mean, one nerve cell, and particularly ones that go from the base of your neck, the base of your skull, I mean, down to your reproductive tract. These have to be very fast acting nerves that get a rapid response down that nerve cell. So conduction velocity. Myelination is probably the most important factor that determines how fast an axon passes an action potential down its length. Because understand, an action potential occurs here or here. So that electricity travels from here all the way down there in an action potential. And the same is true for here. So what's the difference between a myelinated and what's called an unmyelinated axon or nerve cell? In a myelinated, what happens is the myelin acts like an insulator on an electrical circuit. So if you take a piece of wire that is bare, that means bare wire or metal, and stick it in an outlet, and you're touching that wire, the electricity will go from the surface of the wire into you. Now, if you put what's called a non-conductor on it, or an insulator, you can now touch that insulator, which is touching the wire, and the electrical circuit won't go connect through that insulator, and you will be safe somewhat. Depends on the voltage. So with an unmyelinated axon, the action potential has to travel through every millimeter, I should say actually micrometer of length of that axon. In a myelinated, that means what are called the Schwann cells are literally covering part of that neuron. And we do have cells that cover the nerve cell body too, to speed up the neurotransmission. So what happens, guys, is this is kind of hard to explain. But what the myelin does is it allows the electrical current to speed through it without having to activate all those channels and pumps. So that means the current passes through, in effect, without this going through the positive-negative exchange. Don't worry about how that works. But for example, if this is, and I'm just giving you a number, 1/10 of a millimeter long, that neural impulse has to travel 1/10 of a millimeter. If this was 1/10 of a millimeter long, you don't count the whole length. You just count these little slivers, which might be maybe 0.01. That means you tenfolded the speed because you're just counting this as the distance, what are called the nodes of Ranvier. Just understand that occurs. So this is kind of an electrical shortcut that allows electricity in the cytoplasm to pass along without having to go through every positive-negative exchange. They only occur here. So, how does a transmission pass from one neuron to another neuron or to a gland? And this is going to involve some terms because neurotransmissions go in one direction, from the body of the cell to the terminus. So we're going to see a term called presynaptic neuron. That means the incoming information flow is going from here to there. This is neuron 1, this is neuron 2. That means it's receiving a signal from here. This is called the postsynaptic neuron. That means it's receiving a signal from there. This is called the synaptic cleft. It's the gap between these two cells in which these chemicals called neurotransmitters are going to travel from here to there to hopefully, in many cases, what we call an excitatory potential, excite that cell. So just be aware of those terms, particularly when we look at the autonomic nervous system. So the synaptic cleft, it means a gap between communicating nerve cells. It's a fluid-filled space that is surrounded by endoneurium. That means it's protected, it's covered. It's its own little sac, okay, that connects those two cells and has this fluid in it. And this fluid is a very small cavity. Okay, what it does is it allows chemicals in it to diffuse from presynaptic to postsynaptic neurons and allows what's called unidirectional communication. And you'll see this in a minute. So how do neurons connect to neurons? There's three ways. What's called axosomatic. That means the axon of one nerve, the terminus area there, is touching the body. That's what soma means, of a nerve cell. Why do you want to do this? It's just a way to do it. It's a very rapid communication. It's just a way to do it. And then there's what's called axodendritic. That means the axon, the terminus, touches an actual dendrite. And guys, usually in this situation, there's multiple neurons communicating. This allows sometimes what's called graded responses. That means you have three neurons talking to this one, and one neuron might not give enough action potential to do it. It gives a greater potential, which only travels to here. But if two of them are, it might give a bigger stimulus that causes an action potential. And then is what's called axonic, in which an axon actually attaches to another axon at the bare point, and that causes a very rapid communication. Now that's a weird one because the action potential also travels this way and that way. And this could actually make this nerve cell less likely to be stimulated by other neurons. Don't worry so much about that. Just know that these connections are there and they could be very complex. So let us look at a close-up of the synapse. That means this area right here between the terminus of the presynaptic neuron and the surface of the postsynaptic neuron, which could be an axon, the nerve cell body, or a dendrite. So what we see here is that when an action potential comes down here, it's going to stimulate the intake of calcium into the neuron. And that calcium is then going to move these little things called vesicles that store neurotransmitter chemicals of various types. And that's going to force these things along a cytoskeleton to undergo exocytosis. It means the chemicals in here are going to be released into that synaptic cleft, which is, basically right, it's a covering right about there. And then those chemicals are going to attach to what's called a postsynaptic receptor that's going to cause sodium to rush in to the cell membrane right about that little region. So let us look at this step by step. In step one, an action potential travels down here. And we don't care how it was produced. But now what happens is the sodium and potassium are going crazy here. That opens up a special type of channel called a calcium channel. And that allows calcium in here, which is much higher than on the inside, to rush in. And then that causes these vesicles we're going to see to eventually move these vesicles, okay, to spit out neurotransmitter in that cleft. Now the neurotransmitter, if all things are okay, is going to attach these little things called receptors on the postsynaptic neuron. Okay, so there we go. Now at the same time, any extra neurotransmitter we're going to see eventually gets destroyed or absorbed back into the neuron to make sure that each action potential releases only a certain amount of neurotransmitter and there's no buildup. This is the problem with certain toxins. They prevent the neurotransmitter from being broken down and can cause a buildup. And this is what a lot of what are called serotonin inhibitors do. That means like Elavil. Serotonin is the happy, sometimes the happy hormone, we call it the happy neurotransmitter. And if we could prevent it from being taken up, we could actually cause an overstimulus of it that makes people happier. And it can sometimes alleviate depression. But sometimes it becomes addictive, or too much of it can cause literally, almost like hallucinations in some case, they believe. So we get the neurotransmitter again, is doing its job, okay, and this cell is now responding with an action potential. And if there's no more stimulus, the whole system shuts down. So what happens is when neurotransmitter gets in that cleft, what we call the termination, this is only within milliseconds. That means that neurotransmitter, if all things are right, it degrades, it breaks down right away. It gets what we call reuptaked by either astrocytes or the axon terminus itself, or enzymes in this inactive cleft. Just, again, like here, just break it down. And sometimes the neurotransmitter is recycled, sometimes it's not. Now, what is the different circuitry? It means now that we know what a synapse looks like and how it works, how does connection to other neurons work? Because we're going to see this thing called a pathway. Your body is just not one neuron talking to another. It could be several neurons talking to one neuron, or one neuron talking to several, or parallel groups. And sometimes a neuron even talks to itself. And this gets kind of really weird, particularly if you're riding a bus or in the public. Okay, so we're going to look at different what are called neural pathways that make up your body. One type of neural pathway is called divergent. Now guys, pay attention. The information is coming in this way. That means this is the first cell getting excited. Look at that split axon. So what happens is an action potential goes down there, goes down there. If it's enough to get them excited, they split off into two. And so on and so forth. We find these pathways in the central nervous system particularly a lot. Sometimes we see this with muscle, particularly when we're looking at exciting a whole muscle. That means if this neuron is fired, it could actually be responsible for talking to many neurons that excite a muscle group. And right next to it, you could have another pathway that only excites maybe one muscle cell. This is what's amazing about this. You got millions and millions of these pathways in there, all overlapping and doing whatever. This is what makes neurosurgery very difficult, and I'm serious about that to the point where it's one of the most stressful jobs you have, particularly in the brain, where these pathways are incredible and very specific. Then we have what's called a converging circuit. It's the opposite of a diverging circuit. What does it look like? There's several types. So a converging means that what you have here is you have the receiving cell, you have one postsynaptic cell, and three presynaptics. Now this gets to be a problem because you have input from three. And sometimes what happens with this, these are what are called graded. That means this neuron might be responsible for a very critical feature that requires a certain amount of input. So what happens here is that it might produce an action potential only if 2 or 3 of these are working but not one. Then it gives a brief greater potential. It doesn't become an active potential. And sometimes what happens is you want a serial effect. That means number 1 fires, number 2, and number 3. And that adds up to produce an action potential in a certain sequence. This is another way of convergence. Look at this. You have one input, but it has multiple axons that give various output. And this sounds kind of weird, but you can have a greater potential that goes down one axon, and then a full action potential that goes down all of these. And this can then give this cell the ability to have a different response based on different responses from here. Now, my favorite type of circuit is what's called the reverberating or oscillating circuit. And the best way to explain this is not to look at the definition, but just look at it. So here we go. Direction of the impulse. This is your first presynaptic, gets an impulse, sends it to this. This now sends a signal to that postsynaptic one, which is now presynaptic to this one, okay, but also this one. But now look here, and this gets really kind of weird. So this one now communicates to this one, which communicates to this one, which communicates to that one. Now, how do these work? Okay, we didn't mention, you're going to see this a little later, but there are sometimes what happens is one neuron causes excitation to the other. Sometimes a neuron causes another neuron to basically be harder to excite. It takes that action potential, that resting potential, that minus 70, and puts it down to minus 90, maybe minus 100. And it actually inhibits this one from getting a response. So these circuits have different purposes. Sometimes in the most common reverberating circuits, and you find this with like pregnancy contractions stimulated by oxytocin, it causes what you want to happen, because let's say this neuron is causing smooth muscle contraction. So what happens is you want to maintain the smooth muscle contraction by a stimulus comes in. You don't want that to keep, you don't want to keep saying contraction, contraction, contraction. You want those contractions, let's say, just keep occurring. So the circuit keeps feeding over and over and over again, which is probably what makes contractions very bad. Okay, and eventually the circuit can stop. Eventually, when enough, level of signal comes down, and eventually you stop reverberating. But this allows a continuous pathway to ensure a constant and quicker output. So don't worry so much about these, but these are used a lot in memory. They're used in various what we call feedback loops in the body, but very exotic pathways. Now we can look at what's called parallel after discharge circuits. And this is also a bizarre one. This is not reverberating. Notice now. So a signal goes out to 3 parallel circuits that talk to this guy. So what does that do? These are very important in memory. And let me give you an idea of this. Is what happens here is you get input. Let's say you look at an apple. So that apple sends a stimulus to an output of what do I do with the apple? Or how do I interpret an apple when I see it? So what happens is these pathways that are stimulated here, they could be related to other pathways too, that, for example, have to deal with the taste of an apple, the color of an apple, and send all these signals. So what happens is you get one input that could stir a lot of observations about the apple, because these can actually be hooked up to another input, another neuron that says red. So you could equate, so sometimes you'll see a red thing, and this could be the shape of the apple, like saying a ball-like stuff, and that could actually give you the same input as here. And it can actually even stimulate the memory for taste to the point where you even think you taste an apple. So don't worry about this, but this is the complexity of your nervous system. And this is where strokes become a big issue, strokes of the brain. That means infarctions, death of the cells of the brain, because it could affect part of this pathway, parts of these pathway, and cause variations in memory loss, which is really interesting. Now, I mentioned earlier that an action potential from one, the transmission of an action potential from one neuron to another can either excite the postsynaptic neuron to have an action potential, or it could inhibit it. That means somehow shut it down in a pathway. So we have what's called inhibition. That means a neurotransmitter could actually prevent a neuron from firing and stop a firing. This is very important in reverberating circuits. Don't worry about that. And some of the parallel circuits you see. So let's just look at how this works. So let's look at a presynaptic inhibition. There's neuron number 1, which is called your starting presynaptic. There's neuron number 2, which is your first postsynaptic. And then number 3, which is second postsynaptic. So what could happen here is you have this neuron excites that one, that excites that one. How could you turn that off so that this person is not getting a signal anymore? You could have what's called an inhibitory neuron. That means a neuron that produces a neurotransmitter that makes him have a harder time or not even get an action potential. So he can't now stimulate them. So what that means is that somewhere presynaptically, that means before that gap, he's not getting any information saying what the hell is going on. That means presynaptic. Something's happening before he's synapsing. Now, postsynaptic inhibition. Here's your presynaptic, postsynaptic. I'm going to keep this one a little simple. That's your first, second neuron. This is given a signal. But what happens here is that this gets what's called an inhibitory response. That means a neurotransmitter here at the same time or before is lowering this ability to get an action potential. So the way this one works is that, let's say this nerve is firing and this is getting excited. We can shut down that excitation and further impulse transmissions by having him produce a neurotransmitter that basically shuts this baby down. It makes it harder now to have an action potential, so he either has to work harder or any work he's doing is producing a mild grade of potential that goes nowhere. So just please be aware of this. But there's something called an inhibitory setup in which an action potential could actually prevent a postsynaptic neuron from getting information in one of two ways. Okay, and what happens with these inhibitions is, again, it could affect the amount of what's called excitatory or inhibitory postsynaptic potentials, and this can sometimes produce a gradation in a stimulus. That means you feel something happens to you, and you can control the amount of it based on these pathways. Don't worry so much about understanding this fully. But so to kind of review this a little, guys, we can see here, this is a neuron that's just doing its thing. And what happens is it could be attached, and that's a axosomatic attachment, same is true for here. It could be attached to what's called an excitatory neuron, or an inhibitory. So these two are trying to get this guy excited. But he, in a way, gets him excited in a way that it makes it harder to get an action potential. Whereas he's trying to make it produce an action potential. So if he fires without him, you get a definite action potential. If he fires at the same time as him, you get maybe no threshold at all, or it gets difficult to get a threshold. Okay, so we're now somewhat coming to the end of this torture, and we're now going to look at neurotransmitters in general. And I'm not going to make you know all 50 types, and actually there's more. I mean, the more we look at the human body, the more we're finding out that certain things actually act as or assist sometimes with the function of neurotransmitters. So basically, most neurons make 2 or more neurotransmitters, which is really kind of freaky when you think about this, because how does this calcium system that moves the neurotransmitters from one part to another know what to produce? And what we think happens is that it depends on the body of the cell and what type of neurotransmitters it's producing to move these basically vesicles down. So it's a pretty complex thing when we look at this. But some neurons make one, some make two or more, for the most part, types of neurotransmitters. And we're going to see that neurotransmitters can be classified various ways. Aren't you happy about that? Either by their chemical structure or by their actual function. And again, this is because different types of people studied neurons and gave their own way of classifying them. So let us look at the first one. And we're talking about now, when we look at the classes, we're talking about major groups that have subsets of them. So one of the major categories of the amino acids are what are called the acetylcholines. And we usually just say ACH, not ACH. We didn't say that. We just say ACH, capital A, small k CH. This is one of the most common neurotransmitters in the body, particularly in the autonomic nervous system. And we're going to see that this a lot is associated with muscle excitation for the most part when we talk about exciting muscle tissue. Okay, it is one of those neurotransmitters that once it's used, it has to be broken down and recycled. That means reuptake. And don't worry about acetylcholine, transferase, any of these type of stuff, and how it's made. But and there's an enzyme called acetylcholine esterase, which kind of breaks it down. Don't worry about that so much. But we know that people can have various levels or diseases of this or this that affect acetylcholine, production and use. So acetylcholine is just one of those neurotransmitters, and it tends to a lot of times cause excitation. That means it causes postsynaptic excitation. Then we have another class called the biogenic amines, and this is a big category that includes what are called the catecholamines. That's how you pronounce that. And there's dopamine. Dopamine doesn't make you stupid. It's actually one of the more what we call the relaxing type of inhibitory, sometimes neurotransmitters in the central nervous system in the brain. We have what's called norepinephrine and epinephrine, which is also produced by, the adrenal glands. So we get a little connection here between the adrenal glands and the that means the endocrine system and the nervous system here. So these are what we call catecholamines. They could be excitatory or inhibitory, depending on the receptors that they touch. Because guys, for each, and this is going to be a problem, for each neurotransmitter, there can be receptors that respond differently to them in intensity and also whether it causes excitation or inhibits excitation. And then there's serotonin and also histamine. Histamine being a neurotransmitter that's very exotic, usually associated with stimulating pain receptors, because it also has a job in the immune system too. Okay, we see that a lot of these, again, are broadly distributed through the brain, but you also find them in the autonomic nervous system. And a lot of them, like, for example, serotonin, dopamine, play a role in behavior and we think your biological clock. Then we have what are called the amino acid neurotransmitters. These are ones that are up to a lot of debate because these are things that you also take in your diet. And whether diet influences the level of these is up to debate. So that means if you eat a lot of aspartame or glutamine, which can be converted to glutamate, it's debatable. But anyway, these amino acids, again, play very particular roles in regulating and working with other neurotransmitters. And particularly, you find a lot of these in the central nervous system. And then they were what are called the peptide neurotransmitters. That means they're individual amino acids glued together to a small protein type structure. And there's lots of these. Some of these we don't even know yet. We keep finding them over and over. And they're usually responsible for fine tuning and also like communicating signals from organs to another organ and also to the central nervous system. And sometimes these are actually found in, body fluids and could actually be found in the blood in many cases, which makes them almost a hormone, but they do have neurotransmitter effects. And this includes the endorphins, particularly, which are related to the, opiate type compounds and whatever. So again, there's many, many neurotransmitters Then there's a group called the Purines. They're related to ATP, and we now believe ATP even is a neurotransmitter, and a breakdown product of ATP called cyclic AMP is a neurotransmitter. And these are found throughout the central nervous system and peripheral nervous system. And they induce a lot of things, and we believe even may allow neurons to communicate to glial cells and glial cells to communicate to neurons. And then this is, kind of a newer category, what we call the gases. That means nitric oxide, not nitrous oxide. And when we discovered this one, we found out, wow, if we can control nitric oxide, this also influences sexual response in men and women. And it is one of the things that's affected when you look at drugs like Cialis and Viagra. So these are, again, gases that travel through body tissues, travel through the blood, and includes a variety of compounds that are involved in how cells signal each other. And even carbon monoxide, okay, is a neurotransmitter, believe it or not. It's also a toxin too, but it could act as a neurotransmitter. And then there's what are called the cannabinoids. Okay, and this is what allows you to get your thrill out of cannabis, out of, basically marijuana. And these are actually related to vitamins and fats, funny enough. But don't say I'm having my cannabis so I can take my vitamins. And that's not the point of this. Okay, but these activate, again, certain receptors. And this is why we have issues with cannabis, because they are involved in memory and learning and could actually throw that off if they become abused, and particularly in young children. Exposure could affect learning and memory and brain development. Okay, when we look at neurotransmitters functionally now, this is where we say, do they excite? That means, do they cause an action potential, either graded or a full action potential, or do they inhibit an action potential to occur? And I'm not going to drill you on this completely, but just know that here's this thing called GABA, gamma-aminobutyric butyric acid, and glycine. These act in the brain and have usually inhibitory response. And these are used clinically sometimes in people to slow down brain activity when we want to, in cases where you don't want it. And then glutamate is excitatory. And this is a problem when you hear about monosodium glutamate. It's a seasoning usually we associate with Chinese food, even though other cultures use it. You can find it in sugar beets and other vegetable-derived salts and sugars and stuff like that. So we find this in plants a lot. And this can, we believe, can induce vasculature changes that can cause headaches in some people, and in some cases, migraines. Acetylcholine is generally excitatory, particularly to the muscle tissue. But the funny thing, it's inhibitory in cardiac tissue. And this is something people don't pay attention to, because I wrote an anatomy and physiology book that's used at the high school level. We don't use it for our class because it's not as comprehensive for a year-long class as ours. But a person complained to me once when I said how blocking acetylcholine could be used to encourage cardiac beat. And they're like going, whoa, that's not right, because you can stop the heart. And they didn't realize that acetylcholine has inhibitory potential on cardiac muscle. It allows us to control the rate of cardiac muscle, because cardiac muscle, we actually found out, beats faster than you need. So we can actually, and it sounds stupid, slow it down with acetylcholine regulation to keep it either fast or slow, depending on how much acetylcholine is in the system. How do neurotransmitters work? And this is the last of our torture for now, is it varies. Okay, there are what are called channel-linked receptors. That means certain neurotransmitters, they attach to a channel that allows things to go in and out of the cell. That's what we learned about. That's the primary neurotransmitters we learned about. We hid the next one from you. There are also G-linked ones, which means a neurotransmitter attaches to a protein that then attaches to another protein that then sends a signal to another protein that then sends a signal due to changes that protein does, to a channel. This is called indirect. The G means it's linked to what's called a G protein that communicates the signal to here. Why do you want to do this? It just allows different degrees of regulation and allows this channel to be hooked up to, let's say, an excitatory or an inhibitory receptor to control the same channel. Isn't that cool? So on that note, you are experts on the nervous system for now.